So hello, everyone. We are going to be talking about the C9ORF72 effects in ALS and addressing neuroethical relations to ALS diagnosis through a bibliometric analysis perspective. So I am the associate. My name is Knox, and I'm the associate for this project.
Hi, everyone. I'm Akhil Patel, and I'm the co-associate of this project.
Hi, everyone. I'm Navroop Saini, and I'm one of the subsequent authors for this paper.
Hi, I'm Lauren Mansfield, and I'm one of the subsequent authors for this paper.
Hi, everyone. My name is Varsha, Iyengar, and I'm one of the subsequent authors for this paper.
I'm Baul, and I'm also a subsequent author.
I'm May, and I'm also the subsequent authors of this paper too.
Hello, my name is Lillian Searles, and I'm a subsequent author.
Hi, my name is Sherry. I'm also one of the subsequent authors.
Hi, I'm Wendy, and I am also one of the subsequent authors.
Hi, my name is Audrey, and I am also a subsequent author.
So table of contents, first we are going through the intro and objectives slide, then background, methods, results, discussion, and finally the conclusion of our study. So first we're getting into intro and objectives.
When exploring the impact of C9ORF72 mutation on amyotrophic lateral sclerosis, or ALS, progression, it is mandatory to investigate the neuroethical implications within. By analyzing the genetic foundation and the ethical considerations of ALS, we can infer that when treating ALS patients, many scientists and doctors often fail to consider ethics during diagnosis. Our research contributes to a better understanding of the interchange between genetics, neurology, and ethics regarding ALS.
So now we're getting into context and objectives. So the C9ORF72 gene mutation is a huge risk factor in patients with the disease. It causes a hexanucleotide repeat expansion in the DNA, and neuroethics also play a huge role in ALS diagnosis and treatment, including other diseases and medical issues that many will face. Some research efforts have focused on unraveling the intricate genetic/molecular pathways implicated in C9ORF72 ALS, which contributes with contributions being made by many influential authors. Many researchers are utilizing animal and cellular models that effectively replicate ALS pathology in order to assess novel interventions and therapies. And some of our objectives during this project was to create a comprehensive review on the effectiveness of addressing neuroethics and ALS diagnosis through bibliometric analysis, to also address trends in research journals and articles that relate to these issues, and to discover when neuroethical perspectives clash with C9ORF72 gene mutation research and why.
Our problem statement is that neuroethics has often gone unchecked when it comes to disease diagnosis, especially when scientists and doctors believe that if a diagnosis works, it should be automatically used on the patient. Our goal is to highlight the undiscussed gap between neuroethical reasoning and current research on the C9ORF72 gene mutation in ALS and diagnosis methods. The significance of neuroethics lies in the ability to assess the societal and ethical implications of neuroscience, which we will use to further research this topic and its relations to ALS.
So from the study papers we used for our research, we were able to find some senior authors being part of multiple papers with an average frequency of about 2, and some authors have a frequency of 3. However, as an outlier, Dr. Patel is an author for about 4 of the papers we used to conduct our research.
So now we're going into background of our research.
So since we're trying to do a research about C9ORF72 gene and ethical research, we found out that the GGGC hexanucleotide repeat expansion in the C9ORF72 gene is the most common mutation in both familial and sporadic disease form. The genetic mutation is reported to be present in 40 to 50% of patients with hereditary ALS and 5% to 10% of patients with sporadic ALS. Talking about neuroethics, the bulbar affect, PBA is the treatable, but it is really under-recognized neuropsychiatric phenomenon in the ALS. The disorder is stated as uncontrollable laughter and crying. Medication can be managed symptoms, but it may be difficult to receive a diagnosis and treatment, making the patients to advocate for themselves. This slide shows about the top 10 authors with most cited papers throughout the research. The most cited paper goes through 257 citations, and the average goes through 152.
Now we're talking about methods in our research.
Our initial Web of Science search produced 275 preliminary search results. 100 of those were eligible for a study and 75 were not eligible for a study. Of the 100 that were eligible, 95 were articles and 5 were review.
In terms of the method search strategies, we conducted a Web of Science search by narrowing the publications from 1996 to 2024 to analyze any of the evolving publication patterns. And the initial keyword that we used was ALS, which initially yielded 275 results. From there, we narrowed down the search with an interest in the application of ALS to clinical practices and decided to add neuroethics as another keyword. This then allowed us to closely examine the 100 eligible studies with 95 primary articles and 5 reviews. 75 of these studies were considered non-eligible with exclusion criteria that focused on specific regions, demographics, or genetic mutations. And from there, each of the sources, the author, year, article title, journal source title, affiliations, and number of citations were recorded. And this data was then inserted into a Google Sheets to be converted into graphs and figures.
As mentioned before, for the data extraction, the preliminary search results included 275 reviews and articles. The eligible top 100 cited sources were exported from Web of Science to then be filtered into bibliometrics to incorporate reviews, articles, and preceding papers. The articles were then further filtered to be written in the English language. And then, as said before, for each source, the author, year, article title, journal, and affiliations, as well as the number of citations was recorded. And this data was then inserted into a Google Sheets to be made into graphs and figures.
So now we're getting into our results section.
So based on our analysis, we have compiled the figure focusing on the journals with multiple top 100 cited articles. With an average frequency of about 2.85, this figure suggests that the frequency of journals that are not as great as they should be, especially pertaining to our specific discussion that combines ALS diagnosis with neuroethics. Now, specific results for the C9ORF72 gene is that there are 5 known types of dipeptide repeat proteins, which each possibly playing a frontal role in ALS. When the gene isn't working properly due to mutation, it leads to a buildup of DPRs, which can harm neurons and cause toxicity. One promising marker for ALS is a protein called poly-GP, which is made from the mutated C9ORF72 gene. Levels of poly-GP in the cerebrospinal fluid seems to stay the same throughout this progression of ALS. This protein is being used as a way to monitor the effects for certain treatments for ALS. Scientists are using models of microglia cells, a type of brain cell, to study how toxic these proteins are, and then focusing on neuroethics. The evolving field of neuroethics, as it is now being driven by neurotechnologies, comes with controversy of whether it is ethical to use these technologies to enhance brain function beyond normal levels. This can be seen between transhumanists, which are people who support the usage of technology, versus bioconservatives, which are those who don't want to manipulate human biology through this method. And then it brings into the key issues of neuroethics, and how does one define the boundary between treating illness and enhancing normal function? And the key issues in neuroethics of ALS, as who has the agency to make the choices one with ALS faces after disease progresses, and how can the physician clearly communicate how to manage symptoms effectively and preparing for the difficult progression of the disease.
Now we're getting into our discussion section.
While our bibliometric analysis reveals a streamlined discussion of ALS markers, ALS therapies, and neuroethics of end-of-life care. There is an apparent disparity with literature addressing the neuroethical implications of the C9ORF72 gene mutation. The further research of the C9ORF72 is recognized to be finding methods to restore controls of C9ORF72 protein. However, there is a lack of articles surrounding ALS neuroethics as observed by the references provided. Little to no commonly cited sources focalize on ethical involvement in C9ORF72 research. This further emphasizes the need for a greater volume of research on the subject. Aiming to support the QOL and life expectancy of ALS patients, studies regarding the neuroethics of ALS diagnosis should be a priority of research on ALS, as it is key for clear guidelines to be implemented for this field of both research and treatment.
So in conclusion, the development of new methods for monitoring ALS growth has allowed further outlook into the effects of ALS, which can futuristically allow for scientists to expand their methods of combating the disease. With the growth of neurotechnologies, various neuroethical discussions emerge, such as who gains agency in making decisions for further proceedings concerning ALS treatment. And in order to address limitations while researching this subject, research should be predominantly influenced by gathering a large volume of data concerning the neuroethnicalities of ALS diagnosis in order to increase patient life expectancy and quality of life in the future. And thank you.